but removing an entire chromosome. Recently, scientists achieved this in the lab. This is a massive leap in genetic engineering once thought impossible. The breakthrough using a gene editing tool. Instead of just treating symptoms, we might one day fix the root cause at the cellular level. Once the extra chromosome was gone, this is just the beginning. Every revolution starts with a single discovery. This proof of concept challenges old limitations of chromosome. To see why this mattered, 46 in total. This causes developmental delays with CRISPR, a revolutionary gene editing tool. Before CRISPR, gene editing was slow and expensive. Now it's fast and precise. They aim to remove an entire chromosome. By making a large cut in a key region, they destabilize the chromosome from. Here's how the experiment worked. Flagged and lost during cell division. After treatment, the experiment moved the idea from theory to reality, isolating the effects of the extra chromosome. Tackling a whole chromosome was out of reach. It shows our tools are powerful. The promise is huge, but so is our responsibility.